In the previous lecture, we considered how individual organisms have to allocate the energy and resources available to them to different aspects of their life histories, and how the fact that the resources available to an individual are usually finite means that this allocation often involves trade-offs between investment in reproduction and in other life history parameters. Today, we're going to begin a series of lectures in which we'll look at how the availability of energy and resources affects the size and structure of populations of organisms, groups of organisms of the same kind. That is, we're going to be moving our discussion of the significance of energy and resources from the level of the individual to the level of a group of individuals, a population. Now, one important thing we're going to learn over the course of these lectures is, as you might guess, limitations in the resources available to a population are a primary factor in determining the distribution and abundance of any particular population, which is a primary concern of the science of ecology. Let's begin by defining what we mean by a population a little bit more explicitly. Now, when we talked about biological information in the first third of the course, we defined a population back then as a group of organisms that all were capable of interbreeding with each other. In other words, when we first introduced this idea of a population, we were talking about a population as being organisms that all shared the ability to transfer genetic information amongst themselves. Now we're going to change our perspective to an ecological focus, and we're going to consider a population as a group of individuals that all live in the same place at the same time. Okay? Essentially, what this definition is saying that a population is a group of individuals of the same species that all rely on the same set of resources and energy that's available. Now, by and large, this population biology definition of a population really would refer to the same group of organisms in the real world as the genetic definition that we used in the first third of the course. I mean, organisms that live in the same place will breed together, but they also are sharing a common set of resources. Now, at any given moment, any one individual occupies only one spot in an area, right? It's only of one particular age. It only has one amount of reproduction that it's done so far or that it might do in the future. But a population is made up of many different individuals, each of which differ in these kinds of life history traits, depending, for example, on their age, or depending on their sex, or other kinds of differences among individuals. Now, the current sum of the life history parameters of all of the individual in a population is what we refer to really as the structure of that population. Now, the reason we're interested in what we call population structure is because the structure of a population depends on and tells us something about patterns of resource distribution and resource use by that population of that particular species. More importantly, for our present purposes, where we're going to be going over the next couple of lectures, is that a population structure is, describing a population structure is really the first step towards letting us predict if and how that population is going to change over time. Okay, so how do we describe the structure of a population? There are three main characteristics that describe population structure. Let me just list these first, and then we'll go through them in sequence. These three characteristics are the density of individuals in a population, the spacing of the individuals in the population, and the age distribution of individuals in the population. Let's look at what we mean by each of these in turn. A population's density is pretty self-evident. It simply refers to the number of individuals per unit area. Or if it's a population that lives in some three-dimensional space, then the number of individuals per unit volume. The density of a population is a key attribute of that population because, as we'll see, many of the factors that regulate changes in population size depend on population density. How dense the population is will affect how that population changes its size over time. Now, in spite of the fundamental importance and in some ways the intuitive nature of population density, it actually is sometimes very hard to really measure the density of a population accurately. You can imagine, for example, that if we were interested in measuring the density of a particular species of bird, let's say, that lived in some habitat, say a square kilometer of space, what we might think is we could just go out and count all the birds that are out there, and that might be possible to do. But there are a number of difficulties with that simple approach. So, for example, let's say we're talking about a bird. One problem might be that although we've counted all of the birds and considered it in this 1 square kilometer of space, the birds themselves may not actually use all of that space. For example, some species of birds may confine their entire activities just to areas around hedgerows. And so we'd be misleading ourselves about the population density if we divided through the whole space that we measure, because really the functional space is a much smaller space. Now, there's actually a different kind of problem with measuring population density, which is that in many cases it's simply impossible to just go out and count all the individuals. Imagine we were given the task of measuring the population density of some species of fish that lived in a lake. Well, how are we going to actually count all the fish? I mean, there may be hundreds or thousands or tens of thousands of species if it's a large enough lake, numbers of individuals if it's a large enough lake. We can't go out and count each one. So what we'd have to do in that case is come up with some statistical method for sampling individuals, and based on the statistical method, estimate what the population density is. Now, there's another problem with population density, measuring population density, and that is that it's not always clear what we really should be counting. If we're counting, for example, or if we're measuring the density of a population of birds, as we started with, well, then it seems obvious that the relevant measure is the number of individuals. But there are other kinds of organisms where it doesn't seem as though this is necessarily the best measure. Consider, for example, many species of trees, which can range in size from very, very small individuals to huge individuals. Should we count the very smallest individuals the same way we count the large individuals if we're interested in population density? Well, maybe not, because those larger individuals would be using many more resources than the smaller individuals. So instead, in these cases, we might actually want to measure the total mass of a particular species in an area, what we would call the biomass of the species, instead of just counting the number of individuals. Well, how individuals are distributed with respect to each other in the space that they do occupy is can have an equally important effect on determining the effective density of a population. And this is what we refer to when I talked about the attribute of populations that we call population spacing. Right? Spacing in this case might also be referred to as the dispersion of individuals in a population. Now, in a population, we often think of individuals as being dispersed in one of 3 kinds of patterns. For example, we might say that individuals in a population are distributed randomly. And what that means is that the position of any one individual in the population is completely independent of the position of any other individual. We just throw them down in this space randomly. And this would be actually convenient if we just wanted to measure density simply, because we count all the individuals, we estimate the number of individuals, we divide through by the number of space, and we have density. But individuals, more often than not in a population, are not randomly distributed. On the one hand, they may be what we refer to as uniformly distributed. What that means is that individuals occur in the space in which they exist at more or less even intervals. They're spaced out in a very regular pattern. There are a number of reasons that individuals of a population might be spaced out. In animals, for example, this kind of uniform distribution might result from behavioral interactions. Going back to that bird example, for example, many species of birds are highly territorial. They exclude individuals from a region around them. And that exclusion actually ends up spacing individuals in the area available relatively uniformly. In plants, we often see uniform distribution of individuals when we're thinking about kinds of plants that might actually interfere with each other. For example, trees have crowns that would shade each other. So the size of the crown of a tree in a forest might actually determine the spacing of individuals of that species, and do so in a very uniform fashion. Now, by far the most common type of spacing we see in populations is when individuals are clumped, a clumped distribution. What that means is that the position of any one individual depends on the position of another individual. Specifically, it's more likely to be close to another individual than not. Clumped distributions also can arise from a number of factors. For example, in plants, differences in soil types or the availability of particular critical nutrients in soil may cause individuals to clump in a particular region. So if we go into a field, we'll see plants of a particular species only occurring in small areas, all clumped together, because of their dependence on this particular limiting resource. In animals, clumping often occurs again because of social behaviors. In this case, not behaviors that would cause individuals to move away from each other, but behaviors that cause individuals to come into closer proximity because of, for example, the benefits of associating with individuals of the same species, which might be as simple as the ability to find a mate, improving the ability to find a mate. Humans are a good example of a species with a highly clumped distribution, by the way. In our case, we clump because of social reasons. We also clump because of geographic regions. We tend to clump around places that provide useful resources for us. There's no surprise that most major cities historically have arisen around rivers. And we also clump because of economic benefits in our own society. We're a highly clumped species. Now, analyzing patterns of dispersion in a population not only tells us something about the effective density of a population, it also can give us insight into what resources are important for a kind of organism, or how that organism uses those resources, or how that organism interacts with other species in the environment, all of which help us understand something about population structure. Now, the final main characteristic that describes a population is what we call its age distribution. The age distribution of a population refers to the proportions of individuals in that population that fall into different age classes. Whereas the density and spacing of a population are spatial attributes. Where are you in space? Really, the age distribution is more of a temporal attribute, time-oriented attribute. You know, where are individuals across the course of their lifespans? In some species, individuals are born and reproduce and die all in a single season. This happens in some species of insects, for example, or it happens in some species of what we would call annual plants. So in those cases, all of the individuals in a population really would be in the same age class. But in many, many other cases, individuals will live for across several generations, for several years, and therefore generations of individuals of the same species in a population will overlap in time. In such cases, these populations have an age structure, or what you might also call an age distribution, with different numbers of individuals in different age classes in that population. Differences in the age distribution of populations can be a critical factor for determining how populations will change over time, because different age classes usually have different probabilities of reproducing or of dying. Consider, for example, populations in modern human societies. Now, one way we can look at age distributions is to represent them in a graphical form that we call an age pyramid. You can picture an age pyramid as sort of being like a stack of bars. The y-axis of an age pyramid represents the age of individuals, with the lowest part being the youngest individuals going up to older individuals. And we usually bin the ages of individuals in some relevant fashion. For example, if we were making an age pyramid of human populations, we might have the youngest bin being 0 to 5 years old, and then a bin that's 6 to 10 years old, and then 11 to 15 years old, and so on all the way up to the oldest individuals. Now, the x-axis of an age pyramid represents a measure of the total proportion of individuals in the population that fall within that age class. So it's as though we take all of the individuals of the population, determine what age class they're in, and then we'll essentially make a kind of a bar in our graph that the width of which, along the x-axis, represents the proportion of the individuals of that age class, or of the population that fall into a particular age class. Again, you can picture this really graphically as something like a stack of bars on their side, with the width of each bar being the proportion of individuals, and then the subsequent bars going on up being different age classes of individuals. Now, if we look at the age structure of populations in this fashion, in the form of age pyramids, we can get some insight into how populations have changed over time and how they may change in the future. For example, let's consider what the age pyramids of some very different some modern human populations might look like. If we looked at data, for example, from an industrialized European country, a country like Sweden, for example, what we would see is a stack of bars that actually are all more or less about the same width. In other words, the number of individuals in each age class, the proportion of individuals in each age class, is about the same all the way up the age pyramid. In this sense, it's not really much of a pyramid we're looking at. It's more like a column. This pattern is characteristic of a population that has a high survivorship among adults, in other words, a very low death rate in adults, and a steady net rate of birth that approximately matches the death rate. In other words, the number of individuals coming into the population at the young end is about the same as the number of individuals that go out the old end. And so we have a fairly uniform distribution across age classes. Now, if we looked instead at data from a developing country, take a country like Mexico, for example, we might see something very different. The bars at the very bottom of our age pyramid, those representing the younger age classes, would be very wide. And as we went to higher age classes, the bars would become progressively more narrow. In fact, in this case, we would see something that looked more like a real pyramid. This shape of an age pyramid is really characteristic of a growing population. Interestingly enough, a population that has many young individuals who are going into reproductive age, but which has a fairly high death rate, so individuals fall out of this population at a fairly rapid rate, contributing to the narrowing of bars as we go to higher ages. Finally, interestingly, if we looked at data from the United States, we'd see something yet again a little bit different. Now, in the case of the United States, a developing westernized country, we more or less have an even distribution of ages across proportion of individuals across age classes, but actually we see a bulge in the middle of the age pyramid of the United States, a bulge representing what's called the baby boom. It turns out that, as you probably know, the birth rate in the U.S. increased dramatically shortly after the Second World War for about 20 years. During that time, the average size of the American family grew from about 2.5 children per family to 3.8 children per family. Now, the size of the family then tapered off, but the fact was we were left with a whole bolus, a set of cohorts of ages that were much larger in a proportion of the population. In fact, this bolus, this baby boom bolus, moving through the population as it moved through the prime reproductive years for humans, actually contributed to a second little increase, a second little widening later on that we call sort of an echo of the baby boom, a widening of younger individuals, which are the offspring of the baby boom generation. Now, here's the thing to think about here's the thing to remember about age pyramids, and that is different age classes will contribute to the future of a population in different ways, largely because they reproduce at different rates. If we look at human populations, what we're really interested in looking at, if we're interested in how that population will increase over the next generation, is at those individuals that fall in the reproductive years, say from about 15 years old to maybe about 45 years old. Individuals that are younger than that and individuals that are older than that are not as interesting to us from the point of view of asking how that population will change in time. Well, the structure of a population, as I said, reflects patterns of how that population uses resources as well as the availability of resources it has available to it. More specifically, the age distribution of a population reflects different patterns of births and deaths. The link between birth and death rates and the age distribution of a population is a particularly important link because it predicts how that population will change over time. Thus, to understand how a population might change over time in general, we need to know something about the density of individuals in that population, because that often regulates the rate of change. We need to know something about how individuals are distributed, because that affects the way we might interpret density. And most importantly, we need to know something about the age structure of individuals in that population, because the age structure will tell us how many individuals of reproductive age there are in the population. Well, now that we've gotten a general idea about how to characterize population structure, let's look briefly at how a population might change over time, or how we can start to think about looking at how a population changes over time. In other words, what we want to do now is move from a static view of population structure to a more dynamic view. That is, we want to start to ask how we can model what we call population dynamics, the dynamic change in populations over time. Usually populations aren't static, but in fact do change dynamically. In fact, sometimes populations can fluctuate quite widely. Some populations remain relatively stable, but other populations can increase and decrease dramatically over short periods of time. Ultimately, what we want to understand is what factors we could use to predict if and how a population will change. Answers to this question, or understanding these factors, are important to us actually for a couple of reasons. First, on a theoretical level, understanding what factors influence population structure over time will inform us about how biological systems on the level of populations and eventually communities of species are affected by resource availability. Second, there's really a practical issue here, one that we're not going to dwell on now, but we'll come back to later in the course, and that is as the resources of the planet are increasingly stressed, and as they're increasingly modified, in large part because of human activity, we want to know how to predict how these changes in resource availability might affect populations of organisms in order to understand the kind of influence that we're having on the biosphere. Now, we can use information about the current structure of a population combined with information about the life history traits of that particular species to estimate how a population will change dynamically in the future. Let's begin to look at a couple of ways we might get an idea of how to do this. First, as I've said, the knowledge of when individuals are born or die, or really the rate at which individuals reproduce or die in a population, itself provides a considerable amount of information that we can use to determine how a population might change. Births represent inputs to the population, and deaths represent outputs. Now, individuals in the population essentially come and go as they are born and die, but the collection of individuals that make up a population will either change in size one way or another, depending on the relative balance of the inputs to the population and the outputs of the population. Now, the knowledge that births and deaths occur in a predictable fashion, in other words, that we can use average rates at which individuals will reproduce, or average rates at which they'll die, gives us the ability to understand how to look at these predictions. In other words, we can think of this sort of the way insurance companies think of it. If you buy insurance, the amount you pay for life insurance depends on how old you are. Why should that be? Well, it's because the insurance companies know that the older you are, the more likely you are to die, so they charge you more for it. In fact, this kind of information has been used by humans for many, many generations. In fact, the Romans even use these kinds of actuarial tables to calculate differences, for example, in rates of funeral insurance, or to predict how much food should be stored. The statistical study of populations this way, including studying the characteristics of populations like the age and number of individuals of different sexes, as well as patterns of births and deaths and immigration and emigration, this kind of statistical study we refer to as demography, or looking at the demography of a population. Births and deaths, and immigration and emigration, that is, individuals coming into a population or leaving a population by moving from other populations, are what are particularly interesting to us as demographic events, because they will determine the number of individuals of population that we'll find in a population at some future time. The rates at which these kinds of demographic events occur depend on a number of factors that are extrinsic to the population or intrinsic to the population. Extrinsic factors include things like the availability of resources that we've been talking about, but it also may include any other aspect of the environment that influences how likely it is that an individual is to reproduce, of any age class, or how likely it is that an individual is going to die. Intrinsic factors affecting demography have to do with the kinds of life history characteristics that we spoke of last time. For example, we saw in the last lecture that some species might invest more resources into reproduction every year, or might start reproducing earlier, whereas other species might wait to reproduce or reproduce less every year. These kinds of life history parameters obviously will affect the rate at which births and deaths occur in a population. Now, information about the demography of a population often is represented in the form of what we call a life table. And a life table is pretty similar to what the insurance companies use. A life table would simply have as the rows in this table really information about different age groups. In other words, each row in a table will present information about a different age group in a population. The columns in our table, the vertical elements in this table, include information about things like the number of individuals occurring in that age group, the survivorship of individuals in that age group, the probability that individuals in that age group will die or will reproduce, and so forth. Now, usually we construct a life table by taking actual data from a species that we obtain by following what we would call a cohort of individuals through their entire lifespan. A cohort simply refers to a group of individuals all born at the same time who move through the age classes of that population all as a group. And what we do is we start with a cohort, say 1,000 individuals of some species, and ask ourselves at each age group, at each age, what is the probability of individuals in that group dying? What's the probability of individuals in that group reproducing? How many individuals in that group are left? And we follow this age group through their entire lifespan until finally we fill out the entire life history table, the entire life table of this cohort. Once we've got a life table of a cohort, then what we have is a set of estimates of important life history parameters, important demographic parameters, I should say, such as birth and death rate that vary with age. And we can use these parameters now in equations that will allow us to predict how populations will change over time. So really the value of a life table is that it provides a summary of observed patterns of birth rates and death rates and so forth, patterns that we've actually measured from real populations. Now, another common way to look at the demography of a population is in the form of a graph showing survivorship. That is, the number of individuals that are still surviving in a cohort. We call these kinds of graphs survivorship curves. Now, the x-axis of a survivorship curve is a measure of the age of individuals of the population of the cohort that we're interested in, while the y-axis is a plot of the number of survivors remaining in the cohort. This is typically plotted on a logarithmic scale, by the way. These survivorship curves essentially provide a snapshot of age-dependent survival, and as such they provide a good way to compare patterns across different species, or to compare patterns across different populations of the same species. Now, to interpret survivorship data, to look at survivorship curves, it's often useful to compare the observed survivorship in a population to one of three idealized patterns. Now, the first pattern that we take as sort of an ideal pattern of survivorship is what we call a type 1 curve. A type 1 curve represents a case where individuals of a population tend to live out their entire lifespan once they're born. Survival rate is high throughout life until the very end of life. And so we see a curve that remains very high until the very end of the life and then plummets down to some low level. At the opposite extreme, we have what we call a type 3 curve. A type 3 curve is a curve that drops off sharply when really at the left of the graph. In other words, at the very youngest ages, the number of individuals surviving in a population drops to a very low level. But then once that low level is reached, the number of individuals that remain in the population remains constant but low until the very end of the lifespan. This type of curve, by the way, is often seen in organisms that produce large numbers of offspring early on, things like in marine invertebrates or fish. And the survivorship of the very youngest individuals is very low. I mean, a fish might produce thousands of young, most of which never make it very long. But once an individual gets past this critical early stage, then it will survive for a long period of time. Finally, a type 2 curve is intermediate between these two, and it's something that would look really on this particular survivorship curve as a straight downward line from the beginning to the end of the life. This kind of pattern is characteristic of species that have a constant death rate or a constant survivorship rate across life. In other words, there's no change in the probability that an individual is likely to die with age. Now, this kind of survivorship curve is actually characteristic, for example, of many species of birds. Now, early on, when birds are very young, they are very susceptible to dying. They might be eaten. They might not get enough food to live. But if we leave that early part of the life history alone and just look at what happens once birds have left their nest, actually, they tend to have a constant survival rate. They have a type 2 survivorship curve. If they make it out of the nest, their probability of surviving to the next year is more or less a constant throughout their lifetime. Many species, of course, exhibit survivorship curves that are much more complex than these ideal curves. But most real survivorship curves, based on empirical data that we might get from life tables, for example, generally do show type 1 or type 2 or type 3 patterns at different times in their life history. And these patterns will tell us then what kinds of changes in survivorship or the probability of dying is occurring across the lifespan of that particular population, of individuals of that population. Well, things like life tables and survivorship curves gives us ways to visualize factors such as birth rates and death rates that influence population dynamics. What we want to ask next is how we can use this kind of information explicitly to model population growth. And this is the question we're going to turn to in the next lecture.